Okay, let's get started. In the last lecture, we talked about epigenetics therapy, CRISPR screens, and how to use that for combination therapy potentially. And so I think we are kind of done. There is one last lecture on next Tuesday, but you guys can just watch it online. It's a recording of a biomedical fraud that is potentially influencing patients. And it's because of data manipulation at the computational biology level, and it was detected also using computational biology approaches. So it was actually very entertaining, and we'll guarantee you there is a final exam question on that. So make sure you watch it. Yeah, so let's do a module 3 review. So in this third module, we talk about cancer genomics. I mean, I think in general, computational biology techniques are used in many different biological fields, but it just happened, I think, in cancer biology, because recently there's all these new development and therapy. I think all the new technologies and the computational tools are developed gearing towards cancer biology research questions. And so in terms of mutation analysis, we discussed the mutation calls. We didn't actually ask you which specific mutation callers to use, but they're, like at least you are aware of some tools that people use for making the mutation calls. And at the end, you get either a math file or a VCF file. These are kind of files you can convert between each other with slightly different format. But then based on the mutation, you can annotate the mutation, is this mismatch, truncating mutation, or missense mutation or truncating mutation, or some other things. And also you could infer recurrent mutations, which could potentially be drivers of cancer. And so we talk about looking at gain of function mutations of oncogenes and loss of function mutations on the tumor suppressors as potential drivers in cancer. And then we also talk about cancer type specific mutation load. As we mentioned, those cancer types that appears in outside, whether it's facing the air or lung or facing the food in the GI system, and these cancers usually have a later onset and they have more mutations. Whereas the pediatric cancers or tumors that face inside, like brain or blood, they usually have lower mutation load. And also we mentioned that the mutation distribution across the genome is affected by chromatin state, which you could see based on expression of all the genes and also based on replication timing. And these are from just point mutations, but you can also look at copy number variation analysis and using this approach, GISTIC, to look at regions that are recurrently showing copy number gain or loss. So that's mutation analysis. And then we talk about this subtype analysis. And usually the subtypes are coming from gene expression or protein expression from big project like TCGA. You can cluster the samples, right? Recently, when people are looking at DNA methylation, they also detect the subtype of cancers within a cancer type that has very unusual DNA methylation phenotypes. And turns out a lot of these can also be related to the mutation pattern and or viral infection status. And then so when you have big tumor cohorts, for each sample, you can do this single sample gene set enrichment analysis to see whether their expression signature are enriched in some pathways or gene ontology categories. We also briefly mentioned this co-expression network. When you have a big expression cohort, you could look at genes that are always co-expressed for potential modules of gene regulation. But once we have the different subtypes, I think one important thing is you ask whether these subtypes are different, right? So you could look at survival analysis. You could also ask whether the subtypes are different in mutations as well. Sometimes you could say, oh, the biggest difference of this different subtypes is whether the patient has this mutation or not having the mutation. But in each case, if you want to have a subtype, you have to check whether it's clinically relevant. For example, whether the patient will have in the different subtypes will have different survival. And so we talk about survival. So there's censoring. Patient leave the monitoring, either because they just moved or they didn't want to answer calls anymore, you just lost track of them. And because of that, when we calculate, the percentage of patients still alive at that point, we need to keep track of how many patients are still within the study and eliminate those patients who for some reason just left the study. And so for survival analysis, we talk about two things. One is log rank test, and this usually you just have the classes. You don't have the predictive variables sometimes, or the variable is a categorical data. Whereas Cox regression, you can have even continuous values and evaluate many, many different variable effects, like gender, tumor stage, expression of 1 gene, 2 gene, or 3 gene, mutation status, and so on, and looking at many things together. However, when you have so many variables, just need to be very careful. You don't want to have too many in case you have overtraining, okay? So that's kind of subtype analysis. And we talk about targeted therapy and drug resistance. Yeah, so depending on the mutation status, very often you can treat the patient with, instead of chemotherapy that kills all the normal and the cancer cells and give patient very bad side effect, targeted therapy usually target those activated oncogenes, right? And so they usually have less side effect, and they very often target mutations. And so depending on the cancer, depending on the patient, sometimes after a few months or a few years, the patient will develop resistance. And there are a few common ways. One is they create a mutation right at the drug target location, preventing the drug from going to that pocket, enzymatic pocket anymore. The other way, these are called gatekeeper mutations, right? And the other ones are, supposedly this is a gene and there's some on some pathways. And so there could be a mutation either upstream of the pathway, which then use a different way, or it's downstream of this particular gene that you are using a drug. So the downstream get activated, then this drug in the middle, it will not work anymore. And also sometimes there are parallel pathways that can bypass this. So if you're targeting this pathway, a completely different pathway get activated to turn on the tumor growth. And very often what people found is that there are pre-existing resistant mutations. So initially when the tumor is growing, there are many different mutations. When you use the drug, you kill like 99% of the cell, but then there was one that was already in the population, but just a tiny percentage, they were allowed to grow out, and then they will continue to grow and mutate more. And because of this, combination therapy is potentially a better way. You're hoping that if you treat two drugs at the same time, if there are some drug resistance mechanisms, they at least are happening on different cells. For the same cell to evolve resistance against two therapy, the probability would be much, much smaller. And so for combination therapy, we talk about synthetic lethality. And in order to identify that, you can use chemical screens. For example, you treat like 1,000 different cancer cell lines with different compound and see how much the cell is dying or not dying. And we mentioned that for that, you look at the IC50, which is what's the concentration you have to use in order for 50% of the cells to die, right? So the smaller IC50, the more sensitive is the cell to a particular drug. And that's using one chemical against many different cell lines. But the other way is for genetic screens. So we talk about CRISPR screens. This one is for one cell line, we test every single gene, right? All the different genes to see if we knock out that gene, whether that will make the cell grow slower or faster. And so you can use this approach to identify the essential genes or the new, like drug targets for a particular cancer cell line. You can also treat the cell with the drug and compare with the cell without the drug in the CRISPR screen to identify the biomarkers of response and also identify resistance mechanisms and identify combinations. And we mentioned MAGIC for CRISPR screen analysis. And we spent like almost 2 lectures about talking about cancer immunology, which is really very exciting. As we mentioned, a lot of new technologies, new algorithms, and like funding are pouring into understanding immunology or cancer immunology. And so in order to really understand this well, we kind of went through the different computational questions, right? Because mutation call is still important. We need to know what kind of mutations happen in the tumor. And also, depending on the HLA types of that tumor, different mutations will be presented onto the cell surface, right? So this NetMHC algorithm can predict, based on the HLA type, which mutations get presented on the cell surface. We need to look at what cells are infiltrating the tumor microenvironment, and based on the bulk RNA, you can already do deconvolution or gene set enrichment to figure out whether the bulk tumor has more CD8 T cells or more dendritic cells and things like that. And then we talk about immune repertoires. So depending on the TCR, or T cell receptor or B cell receptor in the tumor or in the blood, sometimes it influences the patient response or overall patient immune health. You can sometimes see a bit of that. And then in order to really understand the overall immune microenvironment, which cells get activated or having differential expression or proliferation, people are now using more single-cell RNA-seq to really look at the different cell populations and whether there are differential genes. And they are also looking at ATAC-seq, as we mentioned, this fixed T cell dysfunction versus kind of the early T cell dysfunction, and how immunotherapy might be able to revive the early dysfunctional T cells, but not the very fixed T cell dysfunction. So epigenetically, the cells are responding differently. And recently, people are starting to use single-cell ATAC-seq as well for these immune questions. And in terms of predicting immunotherapy response, we mentioned kind of describe this tight algorithm, but also there are many different biomarkers, right? Mutation load, we want tumor with high mutation load or microsatellite instability. The tumors with high CD8 T cell infiltration is good. T cell diversity might tell us like immune health. PD-L1 level and interferon gamma level kind of tell us whether the immune system made any attempt to attack the cancer cells. And also whether the tumor has other suppressive immune cell types preventing the T cell from infiltrating the tumor. And so these area, I think it's still very, very, like a lot of things are known. And so I would imagine in the coming years, we'll see a lot of updates. And then we talk about cancer epigenetics, right? So previously people look at just copy number variation. They identify some oncogenes and tumor suppressors already, like MYC or HER2. These are identified from copy number. But when people are doing just single, like a sequencing experiment to look at point mutations, the big surprise they found is epigenetic regulators. It was estimated that 1/4 to 1/3 of cancers are caused by mutations in epigenetic regulators. And we mentioned one way is this DNA methylation or SIM phenotype, but there are many other chromatin remodeler, histone modification enzymes that are mutated. And because of this, there are many epigenetic inhibitors developed. Right? So 5-azacitidine target DNA methylation. So it will demethylate the DNA. And this actually creates a viral mimicry because this cancer cell will start to express many repeat sequence, potentially repeat a peptide onto the cell surface, which might look like a virus and can kind of have an immune boost. But recently, there are also other inhibitors, especially those targeting the mutated epigenetic regulators, like HDAC inhibitor, STAT1 inhibitor, EZH2 inhibitor, and the BRD4 inhibitor. Each of them targeted using a very different mechanism. And interestingly, a lot of them can just leave the normal cells intact, and they mostly kind of just hurt the cancer cells, which is because the cancer cells probably not very stable already, epigenetically, not really in a stable state. And so you use another epigenetic drug to push it over the edge. Right? So in order to really understand what those epigenetic inhibitors are doing, people are using differential expression, they're using ChIP-seq for these different factors. They're looking at histone marks in these, whatever these drugs are targeting, what that relevant histone mark is. They're using ATAC-seq, and you might also be using single-cell RNA-seq with or without the drugs, right? And so very often with CRISPR screens, we also find that whenever you treat the cell with a drug, many other epigenetic factors are important, which suggests that using CRISPR screens, you can potentially identify these drug combinations, which combine an epigenetic drug with another targeted therapy or immunotherapy. Okay, so I think that's all we covered in the third module. It's a lot more translational. We are using a lot of the tools that we have learned in the first two modules, but these areas, there are still a lot of unknowns and a lot of new development. Okay, so that's just the third module review.